Thank you for joining us again today on day two of our three-day virtual event. I'm Ryan Piansky. I'm a patient advocate here at APFED. I'm living with eosinophilic esophagitis and eosinophilic asthma, and I've been very fortunate to be able to work with APFED over the last couple decades now, most of my life. So thank you to everyone for joining this virtual research roundup. This program is tethered to our annual conference that we held in person in North Carolina in June. Today, we'll be hearing from several researchers, most of whom have research work supported by APFED grants. Topics today include fresh insights into novel treatment approaches, screening strategies to improve equity, and the role of AI in advancing diagnosis. We'll also spotlight efforts to support young investigators, along with work focused on mast cells and prediction models in eosinophilic GI diseases, and more. It's a full day of discovery, and we're glad you're here to be part of it. Please participate in our virtual table talks that will be open for discussion this afternoon. The topics for today include what's missing in eosinophilic disease research, where we'll explore where patients and caregivers the unmet needs in current research. We'll also have a conversation and connections table where you could stop in to meet APFED's team and part of the community. We can share ways to engage with our community, lead you to resources, or share ways to get involved with research, from enrolling in studies to patient interviews and survey panels. We also encourage everyone to explore the virtual environment. We have the exhibit hall where we have resources for patients and from our partners. We also have our poster hall where we have visual research highlights, and our sessions tab where you'll be able to find recordings from our June event in Raleigh available on demand. I'd like to thank everyone for being part of this experience. We're very, very excited to be doing this again. And I'd like to thank all of our partners, collaborators, and speakers. With that, I will get into our first session where we will be discussing our APFED 2025 Hope Grant Award. As we announced at our in-person event in Raleigh, thanks to APFED donors, we have awarded two $100,000 research grants this year. You heard yesterday from one of our award winners, Dr. Nicholas Hogan, and today grant recipient Dr. Seema Aceves joins us to talk about her new APFED-funded project. Dr. Aceves is a professor of pediatrics and medicine at UC San Diego and director of the Eosinophilic Gastrointestinal Disorders Clinic at Rady Children's Hospital, San Diego. She received her medical degree, her pediatrics residency training, and her allergy immunology fellowship training at UC San Diego. Dr. Aceves' lab studies longitudinal trajectory, clinical impact, and molecular mechanism of tissue remodeling on eosinophilic esophagitis. She is the MRD Endowed Chair in Pediatrics at UC San Diego and was elected to the American Society for Clinical Investigations in 2013. Notably, Dr. Aceves was also the recipient of APFED's very first research grant. That project looked at tissue remodeling in pediatric EOE and how it might be impacted by corticosteroid therapy, which was instrumental work that paved the way for future treatment. We are thrilled to have awarded a Hope Grant this year to Dr. Aceves for her research project titled Cutaneous Dysregulated Lipid Biosynthesis and Cytokine Production in EOE. She joins us today to share a little more about the aims of this work. Thank you for joining us, Dr. Aceves, and congratulations on your award.
Thank you so much, Ryan. It's a real pleasure to be here. And yeah, I want to thank APFED a lot. You definitely kind of spearheaded my career, and I'm always grateful to APFED for what you do for research and what you do for patients. It's an amazing organization, and I'm exceedingly grateful. Shall I go ahead and try and share my screen?
I think so, that would be great.
Perfect. All right. So again, it's really a pleasure to be here today, and I have to commend Ryan on the mouthful of words that are in my title. So my title is Cutaneous Dysregulated Lipid Biosynthesis and Cytokine Production in EOE, and hopefully by the end of this, you'll understand why I'm just super excited to be able to do this. And I'm going to set myself a timer because those of you that know me will know that I will talk to you ad nauseum if I don't have a timer going. So let me do that. So one thing I do want to point out before we get started is that this is a very collaborative study together with Donald Leung's group at National Jewish. So Elena Galeva and Evgeny Berdyshev are really the drivers for the mass spec and the cytokine. And this is really like a nice marriage of collaboration from a group that really started out studying atopic dermatitis and how to diagnose it and how to treat it. So I know you all know about EOE. but just as a reminder, it's diagnosed with an endoscopy with a biopsy. And that's really kind of what I want to focus on, is that you need that endoscopy with biopsy, and there's no way around it. And that is incredibly hard. And I don't need to tell, patients or advocates about how difficult it is to be having to go through endoscopies over and over again. But once you have that endoscopy, the pathologist looks at the histology. You must have that more than 15 eosinophils per high power field, more likely if you've got atopy. And both of those will come back as relevant to the presentation. So EOE, as an allergist who takes care of kids, I really think of EOE as part of the allergic march. And this was shown by David Hill and his colleagues at Children's Hospital of Philadelphia a number of years ago. The EOE can kind of fall in at any point during that atopic march, but that the risks for EOE are much, much higher in patients that have IgE-mediated food allergy, at least if you're a child. Also, if you have atopic dermatitis, allergic rhinitis, or asthma. So like you heard Ryan say that he's got eosinophilic asthma and an EGID, many patients fall into that category of having another type 2 inflammatory disease. So we talked about how you must have an endoscopy with a biopsy to get this diagnosis. And for years, many of us have been looking for surrogate markers in the blood, in the stool, in the sputum. And it's been a rough road, and that we haven't found anything perfect that reflects the esophageal inflammation that could really get us around that endoscopy with biopsy. So I show you this string test because this is now down to a 1-hour test. This was developed by Glenn Fruta and Steve Ackerman a number of years ago and has been validated and is now out in clinical practice. And we can use that in patients that have a known diagnosis of EOE. And it is a way to drop that string down, pull out the biomarkers. And I show that to you because it's relevant to what I'm going to tell you next, which is hopefully an even less invasive way to tell the inflammation in the esophagus. And so that's what this funding will allow us to really query and get an answer to. So what I'm going to talk to you about is skin tape stripping, which is minimally invasive. This is a number of pictures here that show you how this is done. It's basically a piece of tape, much like a Band-Aid, or if you know what Tegaderm is, that holds an IV in place, except it's not quite that sticky. And what you do is you mark the volar surface of the forearm, like you see in panel A. You drop down that tape strip, you put it down, you push it down with a little pressure device that's shown in panel B. Then you pull the tape strip off and you put it down on that piece of paper that you can see on panel C. We do this 20 times repeatedly in the same spot. And even kids tolerate this. They don't mind. They don't cry. And then what you do is you put that tape strip down into a minus 80-degree freezer so that everything stays nice and stable until you're ready to analyze it. What you've basically done by doing this repeated tape strip is to capture the squamous epithelium, so the surface cells and the surface proteins that are living in the skin are captured on these tape strips, and then you can analyze the tape strips. And that's exactly what Dr. Leung and Dr. Galeva and Dr. Bereshev do. They take that skin tape strip and they analyze it. And you can analyze it for a number of things. You need something called mass spectroscopy. So LC-MS-MS is what that stands for, to look at the lipid profiles. You can do something like an ELISA. That is a plate where you can load on a sample and then look for multiple proteins. So say you want to look at inflammatory proteins like IL-13 or IL-33. Those can be captured in this ELISA plate using an antibody and a color reaction. And you can look at the cytokines that are in the skin as well. You can also do things like proteomics off of that skin, which this group has done. I'm just going to remind you quickly a little bit about the molecular pathogenesis of EOE, which is this is a disrupted squamous epithelial barrier. It becomes activated. It starts to make cytokines like TSLP and IL-33 and eotaxin-3. That brings in the T cells, the EOs, the mast cells. They make more inflammatory mediators like IL-5, IL-9, IL-13, IgG, TGF-beta, and that causes this positive feedback loop for the disruption of the epithelial barrier. So that's very much like what happens in eczema, which is the same type of disease, but it's in the skin rather than in the esophagus. And Donald Leong's group has shown that skin biomarkers can predict the development of food allergy in early life. So this is IgE-mediated food allergy. And this started to trigger this concept of, could we use the skin as a marker for the esophagus? So you don't need to memorize this study, but it's just to tell you that in a cohort of children, you can tape strip the skin, send it for these cytokines that are listed down here, things like IL-33 and TNF-alpha. And when you start to put together the lipid profile in the skin, that's what this NS-SARE stands for, plus these cytokines, then you can then generate these very robust predictive values for the generation of food allergies in these children. So the commonalities between the skin and the esophagus that sort of triggered all of this is that they're both a stratified squamous epithelium. They both sort of have their genesis in barrier disruption. These surfaces can be the first line for seeing antigens and pathogens, which brought us to this sort of new hypothesis of a unified epithelial surface. So do the skin and the esophagus look the same molecularly? And then the question became, well, why ceramides? Well, ceramides are these lipids, and they are made up of a sphingosine base and fatty acid chains with varying numbers of carbon. So their names are intensely complex, as is their synthesis, and I am not going to take you through that because I can't even remember all of the details of that, but they are exceedingly important for barrier function. So disruptions in the synthesis of these ceramides are associated with diseases. They can be altered by type 2 cytokines, and ultra long chain fatty acids are especially important for barrier function. So if you don't have those ultra long chain fatty acids, things can get through that shouldn't be getting through. So we did an initial unfunded study of skin tape strips in kids that have EOE. We had 21 kids, regardless of their disease activity state and regardless of having had a recent endoscopy. We were very particular to choose people that had no history of current or past atopic dermatitis or eczema. The reason for that is because we wanted to see a signal that might be aligned with the esophagus rather than aligned with the skin. We had 17 control participants, 10 were from San Diego, 7 were for Denver. We took those tape strips and we analyzed them for ceramides using mass spectroscopy. And in the future, what we want to do is look at cytokines. We did not in our initial study. Our ultimate goal, again, is to use the skin as a surrogate marker for the esophagus. So again, not something to memorize, but I'm going to show you the upshot of that initial preliminary short study, which was that these long chain sphingolipid ceramides were all significantly lower in the skin, so undiseased skin of children that had EOE. In this study, we didn't see a difference between remission and active, but the time between the endoscopies could be anything. It could be like, I think the longest was about 240 days. And so their therapies had been changed. And so we weren't able to see a difference there. But what we did see was very strikingly, the skin of people that have EOE does not have normal lipid synthesis. And so that was the key driver for this application that APFED has funded, which is the hypothesis that skin tape stripping actually can reflect EOE disease activity if we understand the lipid barrier and if we understand the cytokines. And that other hypothesis is that ceramide and cytokine profiles are actually different in active EOE versus remission EOE versus control participants. So we've designed this study that we based power calculations on our initial study. And so what we need are 15 active EOE patients, 15 remission EOE patients, and 12 control participants. All of the remission in this case, we hope are going to be induced by topical therapy. And the reason for that was simply because we wanted to do something that wasn't potentially influencing the blood and everything else, like a diet elimination or a PPI or dupilumab would be doing. We were hoping for something that was hopefully just affecting the esophagus. We're specifically going to get skin tape strips within 14 days of the endoscopy. So we're being picky so that we can look at activity in the esophagus with the skin findings. and aim one, what we're going to do is we're going to look at the ceramides and cytokines in the skin in remission versus active versus health. We're going to compare those groups to one another. And aim two, we're going to compare the ceramides and cytokines in the skin to the histologic features of EOE using the routinely garnered endoscopy, again within 14 days. So we're really excited to start that study. We've actually, I think, already collected a couple of tape strips from our kids. I left time for questions in case you have any. I'm more than happy to answer them. I really, again, want to thank APFED very much for funding the study, and hopefully we'll get some really cool results that we'll be able to share with you in the not very distant future.
Thank you very much for sharing your research. For everyone online, please feel free to submit any questions through the Q&A panel off to the side of the screen. Um, I'll go ahead and start with one question. So personally, I've never had the string test done. I've only had traditional endoscopies, which, as I'm sure you're aware, aren't the most fun thing to be doing regularly. Um, but I've heard in the past that traditional endoscopies are the best way to diagnose EOE, since you're able to get those biopsies and kind of look deeper. Do these string tests allow you to kind of evaluate whether a patient may have EOE, or are these really just for after the EOE diagnosis has already been made?
Right. So that's a great question, Ryan. And right now, the string test is approved only in patients for use only in patients that have known EOE. So it's used sort of to, okay, I instituted a new diet or I tried a topical steroid and I'm feeling better. I'm not sure the histology is better. Let's drop that string down and see if we can find markers of eosinophilic inflammation. Part of the reason to not do it diagnostically is exactly what you said. I mean, I think endoscopy and biopsy will always be the gold standard for EOE. I think there's probably no way around that, right? And I don't know that it's a hard thing, but in terms of care, it may not be a bad thing, right? Because we really want to know what's going on in that organ, and it's rare when we get to actually look in that organ and see what's going on. So the reason you kind of don't want to use it is you don't know if somebody has a stricture, right? And the last thing you want is for something to get impacted. You also want to look at the other features besides eosinophils. I know, Ryan, you are way up on the literature, and so you probably know this, but we're learning more and more that eosinophils are a great surrogate marker, but they're not the ultimate disease pathogenic cell. You knock them out, you still have EOE. There's just not eosinophils with it. You don't help the symptoms. You don't help the transcriptomics, unfortunately, but they're a great surrogate marker. But when you have that first endoscopy, we want to know, do you have remodeling? Do you have fibrosis? Do you have basal zone hyperplasia? Maybe we want to stain for mast cells or T cells or something else. Maybe we want to do transcriptomics. So that string test is very much more like a follow-up study or follow-up way of gauging EOE control.
That makes a lot of sense, and it sounds a lot easier than being sedated every 6 months when you're
Yeah,
trialing new foods.
yeah,
Yeah,
exactly.
fantastic. Well, thank you so much for presenting that
You're
research.
very
I'm
welcome.
excited to see where it goes.
You're very welcome. Thank you again to AppFed for the funding.
Fantastic. Well, next up, I'm pleased to introduce Dr. Sagar Bapat, who joins us today from California. Dr. Bapat is an assistant professor in the Department of Laboratory Medicine at UC San Francisco and a member of the university's Cardiovascular Research Institute. His research focuses on the intersection of immunology and metabolism with an emphasis on engineering regulatory T cells to remodel fat tissue, work that could pave the way for new cell-based treatments for obesity and type 2 diabetes by promoting the conversion of energy storing fat into energy burning fat. Dr. Papat is a board certified pathologist and is dedicated to translating discoveries from the lab into real world therapies. He was awarded a 2-year APFED Hope pilot grant in 2023 for a project that aimed to explore how our diets affect our immune systems to help identify novel ways to treat eosinophilic diseases. And he's here today to share with us some updates regarding this research. Thank you very much for being here. I believe you had some slides to share. Would you like to dive into those?
Yes. Thanks for having me. Let me just figure out if I can share. Sure.
But to start off, while we're waiting for that, can you briefly tell us what your Hope Pilot Grant project entailed and what inspired you to pursue this line of research?
The question, yes, we study in our lab, we study how diets affect the immune response. And eosinophilia is a very specific type of immune response that is driven by this type of inflammation called type 2 inflammation. And we're trying to understand how diets can influence and bias immune responses away to and away from type 2 inflammation. And so that kind of was the idea that can we look at specific types of diets that may promote, or in this case, particular case, inhibit type 2 immune responses and eosinophilia. And that was kind of the idea of the grant. And so we're focusing on high fat diets right now, which is a diet that is that's widely available in the United States. Understanding how that could work.
That's very interesting. So, where are you at with this project currently? Do you have any key findings that you can share with us?
Yes, so we've been studying how the effects of high fat diets generally, and what we noticed is that there are three types of inflammation. Type 2 and type 3 are kind of the ones that are relevant to our, high fat diet. Seems like high fat diet seemed to reduce type 2 inflammation, inflammatory models, and push forward or exacerbate type 3 inflammation. And we realized actually, looking at models of eosinophilic asthma, um, that giving mice high fat diet, and this is all done in mouse studies, pretty sort of basic mouse work, really reduced disease burden in mice. And so what we can say is that that's a pretty clear result. We're trying to figure out now how that's working. And I think the most sort of the last year what we've discovered is that there's a type of cell that really initiates type 3 inflammation and kind of biases inflammation away from type 2. And that happens to be a neutrophil, which is, in the same sort of innate immune component like eosinophilia, but it's a different cell type. And it seems like high fat diets promote neutrophilic sort of infiltration into tissue and bias away from eosinophilia. And the neutrophils are also known to really very cleanly and very dramatically promote type 2 inflammation. So we're discovering that high fat diet has this interesting effect where if you eat a burger or drink a shake, within hours, both in human tissues we're seeing, specifically human skin, but in mouse lungs and skin and other tissues, there's a dramatic neutrophilic migration. And that's something that is interesting because that drives then Th17 or type 2 inflammation and pushes away from type 2. And so we're thinking about this, food is very related, it's something that we can, we eat all the time, eat food all the time. And so does what we eat affect our immune processes? And we're trying to get more sort of medicinal and chemical about this. So what about the high fat diet? What's in this diet that we're giving to mice that's actually causing this? And we're getting closer and closer, but we believe, it's perhaps too early to provide the particular fatty acid we think is actually causing this. But we're trying to get towards that approach where we can actually identify the fatty acid, the component. And if that's the case, we believe we can maybe use this as a strategy to just, take a pill, basically. You don't necessarily have to, oh, there we go. Well, I kind of gave the talk already. Should I
Fantastic.
go
Yeah.
over it?
Are there any visuals you want to flip through
Yeah,
in the slides
sure.
here?
I think,
Okay.
um, yeah, we can go through the acknowledgements, I think. The next slide. I think I just wanted to mention, our lab basically, this is the intro, that we look at how diet affects immune responses, and we kind of conceive of there being three types of immune responses. That can be essentialized looking at a T cell, and this is looking at a naive T cell becoming possibly three different states: Th1, Th2, Th17. And what's important here is that each of these cell types, they produce different cytokines. And the success of modern immunology over the last 20, 30 years has been that we've identified these particular cytokines, and then we have biologics to actually perhaps target these, and that can have dramatic responses in many immunological diseases. And Th2 also produces IL-5, which is also critically important for eosinophilia. And so what we basically discovered was a high-fat diet. I think you can maybe scroll through a few more slides. And this is, I guess, an important slide. So what we discovered was that, before looking at diets in particular, we were focusing on not just the diet, but lean versus obese mice. And the idea being that obesity is sort of a big public health challenge. And can we shift type responses by just making a mouse go from lean to obese? And we discovered that if you give a mouse a classic Th2 immune challenge. In this case, it was atopic dermatitis, eczema. In the lean mouse, you get a very classic IL-4, IL-13 driven T cell response. But an obese mouse, which is basically a mouse given high-fat diet for long periods, that response becomes Th17 oriented or type 3 oriented. And that's what sparked the idea that maybe the diet itself can kind of shift immune responses, and that can be used actually in a helpful way. And so, and that this kind of also was sparked when we did the next slide, I believe, that actually a disease that like eosinophilic asthma, and this is just showing we measure that in a mouse by looking at airway resistance. And basically the blue line is the airway resistance that occurs in lean mice when you give them eosinophilic asthma, and it's pretty high. And in comparison, mice that were given high-fat diet, the red line, had dramatically lowered resistance, and so they were better off. They were not doing as badly as mice that were lean. And this was really the inspiration to study this more carefully. And so I think I'll sort of skip through a couple of slides and just go to sort of the last keep going, please. There's just more. This is actually yeah. Keep going, more slide. This is actually the interesting part that I was discussing, that we were tracking to identify, what cell types seem to actually go up in tissues, in lungs or in skin, just after eating the shake or eating the diet. So these mice aren't fat. These mice have just been on high-fat diet for, um, 2 or 3 days, up to a week. And we asked the question, well, can we track immune cells? And it turns out that if you look at neutrophils in particular, which is a type of immune cell, that both in the ear, and this is skin basically, and this is the lung, and other cell types, when you give mice high-fat diet for just a few days, in this case it was a week, there's a dramatic uptick in HFD's high-fat diet in neutrophils. And once you stop eating high-fat diet and you go back to low-fat diet, the neutrophils come back down. And so this suggests that there's a very temporal and very immediate neutrophilic response into tissues after you eat a burger or drink a shake. And now we're getting into understanding what the implications of this could be. And so the next slide, please, is really our idea of what's happening, which is that you drink your shake or you eat your high-fat diet, your burger, and there's this dramatic migration of neutrophils into tissues. And we're trying to understand how that's happening, what enzymes and what genes are causing this to occur. And then we have shown that this increased neutrophilia, particularly these are great sources of a cytokine called IL-1 beta. They seem to secrete IL-1 beta and drive Th17 cells. And Th17 cells actually are very potent counter-regulators of Th2, or type 2 immunity. So we believe the mechanism by which high-fat diet suppresses and reduces some disease in eosinophilic asthma in mice given a high-fat diet is that they sort of increase Th17 and type 2 inflammation and decrease type 2 inflammation. And this type 2 inflammation is what's causing disease in mice. So our idea really is, can we use diets as common food that we eat all the time to actually modulate immune response in very potent ways? And maybe that'll help us manage our own inflammatory processes in rational ways. So that's kind of the yes, that's the presentation. I think I have a couple conclusion slides. Yeah, so that's what we're trying to figure out. How is high-fat diet? What is the component of high-fat diet that we're looking at that can suppress this Th2 response in asthma? And there's one more. And the cell types driving this, yes, but both are just kind of the mechanistic understanding. And if you can figure out the cell, if you can figure out the component in high-fat diet, maybe we can create a pill with a fatty acid to take as an adjuvant with the other therapies. And if you can figure out the cells driving this, maybe that can be targeted also with biologics. So these are kind of where we're headed. Sorry about the hiccup, everyone, but yeah, hopefully that was still a reasonable presentation, coherent presentation.
Yeah, no, that was fantastic. And I think we have a couple minutes for questions. So if people in the audience do have any questions, feel free to put those in the Q&A tab off to the side. While we're waiting for some of those to come in, I did want to ask, I think many of us with EOE are very familiar with dietary therapy, managing what we eat to manage our EOE inflammation. And it sounds like this is a very different sort of approach to that. So, how might this research eventually impact the way that we understand or treat eosinophilic associated diseases?
Well, I think when I'm always careful to say that when we do things in mice, we have to validate and validate before we kind of make real suggestions to patients. But if you're just kind of think of it aspirationally, what kinds of how this kind of work could sort of be used by patients. I think it's the idea is if we really develop robust understanding of how very specific nutrients can affect um, uh, immune processes in humans with trials. I think this could be dramatic because I think um, we don't really always have a great understanding of how what we eat affects us. And by even having some concrete understanding, that could perhaps affect how exacerbations of disease. If you know you're vulnerable to a specific type of inflammation, maybe and you realize certain foods can maybe drive inflammation, maybe you've been given recommendations to not eat those foods or sources of foods that have that kind of particular chemical or constituent. So that's where that's what I imagine what I'd like to do in the next during my career, and especially this sort of inquiry and this line of inquiry.
That makes a lot of sense. Yeah. We had a question come through in the Q&A from Jody. So you talked a lot about these like high fat diets and these mice produced a different immune response. We had a question come in about how healthy is a very high fat diet? Is that the one outcome that you can have just eating a very high fat diet to moderate your immune system?
Yes, I mean certainly, a high fat diet. The diet we're giving um, is associated with obesity and weight gain and metabolic syndrome. Um, but I think so. I wouldn't suggest anyone right now go out and start just eating, fast food all day long or particularly fatty meals. And these are all animal fat based diets. But I think the idea here is that this is in small transient doses. So perhaps it could be the case that everyone. I mean, not everyone, but I do certainly eat high fat meals every now and then, and more pretty often. Um, and so there could be an idea that maybe you could eat a meal or here or there to suppress certain inflammation. And I think that's why we're getting away from trying to actually go from the diet to actually finding sort of the specific chemical in the diet that's causing this effect. Because we can identify that, we could perhaps make that into a pill as opposed to asking someone to um, eat an entire meal that's fatty.
That makes a lot of sense. And you gave a little bit of a slide on what your next steps are. So just to follow up on that, do you have plans to build on this work through additional studies or funding opportunities? What's coming up next for this?
Yes, we do have some actually candidates we believe are actually the constituents in the diet, and we identified maybe the sort of the gene pathway that's allowing these constituents to actually metabolize into things that actually allow neutrophils to enter tissues. And so we really are moving forward trying to understand the specific constituent, and if so, we can have a target, and that's something then clinically actionable item that we could actually perhaps consider doing scaling up to from preclinical studies to maybe even something that's a could move into clinical trials many years away.
That sounds very interesting. I'm excited to see where this research goes. Thank you again for sharing, and thank you to the APFED donors who supported this work.
Thank you, everyone.
For those online, we invite you to stop by one of our virtual table talks. Those will be open until 1:15, and at 1:15, we will be starting back up in session two under the sessions tab. So everyone, get up, stretch, join those virtual table talks, and we will see everyone shortly.